learners, welcome to Crown Biology classes. This is Rohit here, and today I will be discussing the part 2 of chromosomes. And I will be covering the topics which are as follows. The first one is genes are located on chromosomes, the second is karyotype and idiogram, and the third is material of the chromosomes. So first of all, starting with the first topic, that is genes are located on the chromosomes. In modern terminology, Mendel's factors are called genes. Mendel called heredity units as factors, and these Mendel's factors are known as genes. We know genes are the units of heredity, and these are responsible for the characteristic features. Genes carry the information that determines our traits or characteristics, which are the features or characteristics that are passed on to us or inherited from our parents. Each organism has very few chromosomes and far more number of characteristics. And if we see, each organism is having very few chromosomes, but the organism is having far more number of genes, and the genes are responsible for controlling these characteristics, or also known as traits. For example, human beings have just 23 pairs of chromosomes which carry thousands of genes controlling these characteristics. Parents pass on traits or characteristics such as eye color and blood type to their children through their genes. And likewise, some health conditions and diseases, these can also be passed on genetically too. Sometimes one characteristic has many different forms. And it is estimated that there are about 30,000 genes in a human genome. And what is a genome? A genome refers to one complete set of chromosomes. So a human genome contains 23 chromosomes. We know there are 23 pairs of chromosomes, in total 46 chromosomes present. And a genome refers to one complete set of chromosomes, that means a genome contains 23 chromosomes. Each chromosome consists of a number of genes. For example, a human chromosome, we know it contains thousands of genes, and these thousands of genes are responsible for controlling the characteristics. Each gene is present at a specific position on a chromosome called its locus. So there is a specific position on a chromosome where the gene will be present, and that specific position is termed as locus. Each chromosome is made up of a highly coiled DNA molecule which has individual genes present along its length. We know that each chromosome is made up of DNA that is tightly coiled many times around proteins, and those proteins are called histones, and that support its structure. So histones are basic proteins, and their positive charges allow them to associate with DNA, and DNA, as we know, is negatively charged. A gene is a short segment, that is a length of DNA, or a gene can be defined as the basic physical and functional unit of heredity. As you can see in this picture, there is a cell in which nucleus is present and one of the chromosome is enlarged, and this chromosome is having the DNA content in it. This is the DNA content, and this is a short segment of DNA which is termed as gene, and there is a specific position on this chromosome on which this gene will be present, and that specific position is known as locus. Now I will be discussing the topic, that is karyotype and idiogram. All the members of a species of a plant or an animal. So all the members are characterized by a set of chromosomes, and those set of chromosomes do possess certain constant characteristics. And the characteristics possessed by the set of chromosomes include the number of chromosomes, that means how many chromosomes are present, their relative size. When we do compare the sizes of the chromosome, Position of the centromere, where the place of constriction will be present on the chromosome, length of the arms. The next characteristic is secondary constrictions. So secondary constrictions are the constricted of the narrow region that are found at any point of the chromosome other than that of centromere, and the last characteristic is satellites. Chromosomal satellite, it is the term given to that part of the end of a chromosome that is separated from the rest of the chromosome by a secondary constriction. Now I will be defining the term karyotype. So the term karyotype has been given to the group of characteristics that identifies a particular set of chromosomes. The characteristics that I have mentioned just now, they will be identifying a particular set of chromosomes. And a karyotype is the preparation of the complete set of metaphase chromosomes in the cells of a species or in an individual organism. And those metaphase chromosomes are sorted by length, centromere location, and other features. During metaphase of mitosis or meiosis, the chromosome condense and they become distinguishable as they align in the center of the dividing cell. So metaphase chromosomes are used during the karyotyping procedure that is used to look for chromosomal abnormalities. And a diagrammatic representation of a karyotype of a species is called idiogram. Idios, that means distinctive, and gramma, that means something written. So ideogram is a diagrammatic representation of a karyotype showing all the morphological features. That means the properties related to external structure of the chromosomes grouped on the basis of the position of centromere, and these are ordered in a series of decreasing size. Or this ideogram can also be defined as a diagram or a photograph of chromosomes of a cell arranged in homologous pairs and in a numbered sequence. Generally, in an ideogram, the chromosomes of a haploid set of an organisms are ordered in a series of decreasing size. For example, if we take the human chromosomes, there are 23 pairs of chromosomes, and if we take the haploid set, that means 23 chromosomes. These chromosomes are arranged in the series of decreasing size. So this is the picture of ideograms of human chromosomes. As you can see, there are total 22 chromosomes which are known as autosomes, and these are arranged in the series of decreasing size. As you can see, as we move from this first to the 12 and then further 13 to 22, the chromosomes are decreasing in size. The last pair of chromosomes, that is known as the allosomes or the sex chromosomes, is represented by XY in case of male and XX in case of female. A karyotype of human metaphase chromosomes is obtained from where? It is obtained from their microphotographs. The individual chromosomes are cut out of the microphotographs, and those chromosomes are lined up by size with their respective partners. As you can see in this picture, this is the karyotype of the human metaphase chromosomes. These are the homologous chromosomes, and this chromosome is having its respective partner, and these are arranged in the order of their decreasing size. And the last pair of chromosomes, that is known as the sex chromosomes or allosomes, this is represented by this pair. Now I will be discussing the uses of karyotype. The karyotypes of different species are sometimes compared. For example, if we take two different species and if we do compare the karyotypes of two different species. And you know what karyotype is? So karyotype is the actual picture of the chromosome set of a cell arranged in homologous pairs and in a series of decreasing size. And when we do compare the karyotypes of the two different species, that means the chromosomes of the two different species, and if we find similarities in karyotypes, Those similarities in karyotypes are presumed to represent what? Those are presumed to represent evolutionary relationship. And what evolutionary relationship is? The organisms that share similar physical features and genetic consequences tend to be more closely related than those that do not. So the features that overlap both morphologically and genetically are referred to as homologous structures. And where do the similarities stem from? The similarities they stem from the common evolutionary paths. So karyotypes also suggest primitive or advanced features of an organism. So just by looking at the chromosome structures, we can predict whether the organism possesses primitive or advanced features. So accordingly, a karyotype may be symmetric or asymmetric. If a karyotype is exhibiting primitive features, then it is a symmetric karyotype. And if the karyotype is exhibiting advanced features, then it is asymmetric karyotype. And a karyotype exhibiting larger differences in smallest and largest chromosomes of the set. See, if we take one karyotype and if we do compare the smallest and largest chromosome of the set, and if they are exhibiting much larger differences in their structure, then it is known as asymmetric karyotype. And this asymmetric karyotype should also contain fewer metacentric chromosomes. And what these metacentric chromosomes are, that you know very well, in which the centromere is located centrally. In comparison to a symmetric karyotype is shown by pinus, that is a tree, and asymmetric karyotype is shown by ginkgo biloba, that is also a tree. So this asymmetric karyotype is considered to be a relatively advanced feature as compared to the symmetric karyotype, which is exhibited by pinus. Now this is the picture showing symmetric and asymmetric karyotypes. The first example is of pinus, in which it is showing symmetric karyotype. And the characteristics of symmetric karyotype are, it shows lesser differences between smaller and larger chromosome in a set, and it have more metacentric chromosomes in which the centromere is centrally located, and symmetric karyotype have no relatively advanced feature. And the example, as you can see, is pinus in case of symmetric karyotypes. And in case of asymmetric karyotypes, the example is ginkgo biloba. And the characteristics of asymmetric karyotype are, asymmetric karyotypes show larger differences between smaller and larger chromosome in a set, and it have more acrocentric chromosome. So acrocentric chromosome, as you know, is a chromosome in which the centromere is very close to one end, and asymmetric karyotype have relatively advanced feature, and the example is ginkgo biloba and flowering plants. Now I will discuss the material of the chromosomes. The material of the chromosomes is the chromatin, depending on their staining properties. That means whether the chromatin will be lightly stained or darkly stained. The following two types of chromatin may be distinguished in the interphase stage of mitosis in the nucleus. So the first type is euchromatin. In euchromatin, the portions of chromosomes that stain lightly are only partially condensed. So there will be certain portions of the chromosomes, those portions will be lightly stained and they will be partially condensed, and the chromatin is termed as euchromatin. The chromatin is considered genetically active chromatin. That means this euchromatin is active for transcription. That means it can be transcribed. And since it has a role in the phenotype expression of the genes. And if it can transcribe, that means it will play its role in the phenotype expression of the genes. In euchromatin, the DNA is found packed in 3 to 8 nanometer fiber. The DNA is found loosely packed in 3 to 8 nanometer fiber in case of euchromatin. The second type of chromatin is heterochromatin. The portions of the chromosomes that are darkly stained, they remain in the condensed state, and the chromatin is known as heterochromatin. Unlike chromatin, this heterochromatin will be inactive for transcription. That means this heterochromatin cannot be transcribed. In 1928, Hitz defined heterochromatin as those regions of the regions of the chromosomes that remain condensed during interphase stage of mitosis and early prophase stage of mitosis, and they form the so-called chromocenter. So those regions that remain condensed form chromocenter. Heterochromatin is characterized by its especially high content of repetitive DNA sequences, as these DNA sequences will be present in high content in heterochromatin, and it contains very few structural genes, and these structural genes are the genes that encode proteins. It is thought that in heterochromatin the DNA is tightly packed in the 30 nanometer fiber, whereas in euchromatin the DNA was loosely packed in 3 to 8 nanometer fiber. These repetitive DNA sequences, these refers to the DNA fragments that are present in multiple copies in the genome. For example, telomeres are the DNA repeat sequences. As I have discussed about heterochromatin, now I will be discussing types of heterochromatin. What are its further types? In an interphase nucleus, usually there is some condensed chromatin. We know heterochromatin are the condensed chromatin, and these condensed chromatin are present around the nucleolus. Then they are known as perinuclear chromatin, and some of this chromatin is present inside the nucleolus. Then it is termed as intranuclear chromatin. Both types of this heterochromatin appear to be connected. This perinuclear chromatin as well as intranuclear chromatin, these are connected and they remain together, and collectively they are referred to as nuclear chromatin. Dense clumps of deeply stained chromatin often occur in close contact with the inner membrane of the nuclear envelope. When these heterochromatin are associated with the inner membrane of the nuclear envelope, that is with the nuclear lamina, then it is termed as condensed peripheral chromatin. Between the peripheral heterochromatin and the nuclear heterochromatin are the regions of lightly staining chromatin, and that lightly stained chromatin is called dispersed chromatin. This heterochromatin has been further classified into the following types. The first one is constitutive heterochromatin. In such a heterochromatin, the DNA is permanently inactive. That means the chromatin will not be transcribed. The transcription is inactive and it remains in the condensed state throughout the life cycle. The most common type of heterochromatin occurs around the centromere. Centromere, as you know, it is the constructed region of a chromosome in the telomeres, and these telomeres are the end of a chromosome, and this telomere is made up of repetitive sequences of non-coding DNA that protect the chromosome from damage, and in the C bands of the chromosomes. So these are the places where this constitutive heterochromatin will be present. In Drosophila virilis, this is a fruit fly. Constitutive heterochromatin exists around the centromeres. So around the centromeres, the constitutive heterochromatin will be present, and such pericentromeric heterochromatin occupies such a large amount of the chromosomes, about 40% of the chromosomes. In many species, entire chromosomes become heterochromatic, and those chromosomes are called B chromosome, satellite chromosomes, or accessory chromosomes. Such chromosomes comprising wholly constitutive heterochromatin occur in cone, that is maize, many phytoparasite insects and salamander that is an amphibian. So these are the examples where this heterochromatin will be present, in which the entire chromosomes become heterochromatic. Constitutive heterochromatin contain short repeated sequences of DNA called satellite DNA. And the satellite DNA consists of very large arrays of tandemly repeating non-coding DNA, and satellite DNA is the main component of functional centromeres, and it forms the main structural constituent of heterochromatin. The DNA is called satellite DNA because upon centrifugation, this is a specialized technique used to spin samples at exceptionally high speeds, and this technique uses ultracentrifuge, which is a sophisticated and advanced centrifuge that operates at an extremely high speed and separates small molecules that cannot be separated from the traditional centrifuge. And through this technique, the main component of DNA is separated. The second type of heterochromatin is facultative heterochromatin. Such type of heterochromatin is not permanently maintained in the condensed state. That means it will not remain in the condensed state permanently. Instead, it undergoes periodic dispersal. And during that periodic dispersal, it is transcriptionally active. That means that chromatin can be transcribed. The best known case is that of the X chromosomes in the mammalian female, one of which is active. As we know, the mammalian female has two X chromosomes. So one of the X chromosome is active, that means in which transcription is possible and it remains euchromatic, whereas the other is inactive. The other sex chromosome is inactive and it forms that interface. What it forms? The sex chromatin or bar body, and this bar body is named after its discoverer, that is Canadian cytologist Murray L. Barr. And this bar body contains DNA which is not transcribed and is not found in males. We know in case of males only one X chromosome is present. So the bar body contains DNA and that DNA is not active for transcription. So these are the two staining techniques. The first one is hematoxylin and eosin stain, and the second one is EM staining technique. In hematoxylin and eosin stain, we can see this euchromatin, it is lightly stained and therefore it is transcribed easily. But this heterochromatin, it is darkly stained and it cannot be transcribed easily. Likewise, we can see in EM staining, this is the heterochromatin that is darkly stained and it cannot be transcribed. And this is euchromatin which is lightly stained and this can be transcribed very easily. So ending this video on a positive note, this is a motivational quotation by Walt Disney which says, "All our dreams can come true if we have the courage to pursue them." If you like this video, then please do like, share and subscribe, and don't forget to press the bell icon so that whenever I do upload a new video, you may get notified. Thank you.